Being a biologist or entomologist is a person who at some point of his life fell in love with insects and made a job out of it. That's actually what I do for the living, investigating insects. While what I'm talking about is relevant for all kinds of small objects in the macro photographic range, most of the examples I will talk about and show you are insects. I will talk a little bit about technology and about methods that we can use to generate digital twins, 3D digital representatives of small objects. One way to do that is photogrammetry. We can also use Gaussian splatting. We start with taking multiple photos from an object from multiple directions. Based on this, we use an approach that is called structure from motion. Estimate the camera parameters, where our camera was with respect to the object, and then we calculate the point cloud. With the point cloud, we can go in two directions. Either calculate a surface mesh and then use this mesh, which consists of polygons, to apply photographic texture extracted from the original images. Then we have a textured polygonal surface model. Or we can try to mimic the visual appearance of an object by calculating anisotropic Gaussian splats and having rendered splat images. This is not really a model, this is more a visual representation. Photogrammetry is not very complicated. All you need is an object. Like here, I put this model of Grauammer. It's a little bird from our collection, and I put it just on a turntable. I used my smartphone to take a couple of images from different angles, 60, 70 images or so. Not too many, but some. And then I used these images just to calculate and generate a 3D model. And the whole process was done in less than 2 hours on a normal computer. We want to extract from that object special features that might help us identify parts of the object. And to make this a little more clear, for example, when you're on a hike and you see a tree standing away 200, 300 meters, you will immediately say that's a tree. You will use characters that your brain uses to identify this as a tree. If you're standing right in front of the stem 10 centimeters, you will also be able to say I'm at a tree, but your brain will use completely different characters and different aspects of the tree because it will see the bark and know the bark and so on. Depending on where you are, you will use different features that characterize this tree to identify it. And to mimic this with a piece of software, you use the image of an object and enlarge it and shrink it. We apply different amounts of blur to the image. The enlarging is the distance and the blur is just the amount of information you can use from the image. And then we look at every pixel of this blurred and size reduced images and compare every pixel with all the neighbors, the larger, the smaller, the sharper, and the more blurred. Whenever this single pixel has a value that is higher or lower than all the neighboring images, then this is a potential candidate for a feature that might be recognized later on. It's possible for every point to generate a key point descriptor. We have this key point and we can describe it regarding the neighborhood of this feature. And with simple words, that means in every direction of this feature point, the image will have a gradient of brightness or grayscales or color or whatever. We can make brighter or darker. The advantage of key point descriptors now is that they are always the same, independent of size and orientation of the object. They are invariant to scale and size. This was an important paper published in 2004, Invariant Key Points from David Lowe. The intention was to be able to identify objects. He took a photo of this plastic toy train and plastic frog, and this was the only training data he used. One image, and he built up a database with his key point descriptors. Here is a part of the train hidden, here is a train but with another orientation, just little parts of the frog. The software was able to identify all three objects and to find them based on these specific key points in the object. This was meant for object detection at the very beginning. And now the question is, how can we use these feature points in 3D modeling? Because that was not the initial intention. I took a couple of images from a common kingfisher, a skeleton, and there are some blue dots which indicates feature points. When you look from different angles, then the spatial relationship of these blue dots differs. Some become closer, others are larger, the distance is larger. The 2D arrangement of these blue dots to each other, they differ. The cameras of your smartphone follow this pinhole camera models. That means that all the visible rays cross at some point and meet behind the sensor. When we know that we have such a pinhole camera and we take images from different sites, then there is just one solution how these feature points are arranged in space. We need images from different angles of an object. We need to know the parameters of the camera. Then there's just one solution where these actual points, red here, lie in the 3D space. I took 48 images just on one plane around this skeleton. This is the projection of the actual points in a 3D space. An image of an insect. All these dots are feature points that the software identified and said, this point I can find again on an object because it's independent of space and scale. Whenever you show me an object that has this set of feature points, I know it's exactly this. When we compare two images that are very close to each other regarding where the camera was positioned, then we have a high number of overlapping feature points. The blue ones have a high confidence, the red ones have a low confidence. When we compare images from very different angles, of course, there will be much fewer feature points that can be detected and connected in the two images. It becomes clear we need a lot of images from different angles to be able to project as many as possible points in the 3D space to use that to generate a model. Insects are specifically problematic because they're on macro photographic range. We have transparent structures like wings. We have structures that are very reflective. They reflect light. Large brightness gradients, so some parts are very dark, others are very bright. We have hairy structures, very dense hairs, for example, and many of the structures are complex and very fragile. But most important, there is this reduced depth of field. You will always end up that just a tiny part of the object on the image will be sharp and the rest will be blurred. That's because you have just a shadow depth of field. But what we want, and of course what we need, is an extended depth. We need the whole object to be completely sharp to be able to compare these feature points. One thing you can do is take manually one image after the other and combine them to a single sharp image, which is done like this. There's commercial software, free software that combines image stacks to one sharp image. A couple of smartphones do that even automatically. They have a function of extending the depth of fields. But the issue is the result will follow more a telecentric lens and not this pinhole camera model. This is what we need actually, and this is what we get. You see here are two cubes and they have the same size, but one is standing a little bit further away from the camera and the other one is a little bit closer. When you use a normal camera, you will see these two cubes are different in size. There are two explanations for that. The first is the right cube is smaller than the left one. The second explanation is they are of equal size, but the right cube is standing further away from the camera than the left one. From one image, you will not be able to know what of these two scenarios is correct. When you have a telecentric lens, they look as if they have the equal size. For example, when you use a camera in quality assurance, in the production and so on, and you want to measure the size, but you don't want to care about how far the object is away from the camera, you will use such a telecentric lens. But if you want to do a 3D reconstruction, we always need this pinhole camera model. We need to know when this cube is smaller than the left one, that it's standing further behind, because otherwise we will not be able to project these feature points to their correct position in space. We developed this specific scanner for small objects, and we called it the Darmstadt insect scanner because it was developed in Darmstadt, and the initial purpose was to 3D scan insects. Disk 3D. We published this as an open source project, first of all, but there was so overwhelmingly much feedback from museums and institutions that wanted to have it but were not able to really build it, that finally we funded a startup which is now further developing this scanner and selling it to the market. The principle is we have the insect mounted on a needle. That's how insects are stored normally. We have a couple of motors that turn around the insect. We have the camera mounted on a rail. We will take movies of the insect and extract from every image just the sharp plane. This will be done fully automatic. What we take is a stack of images from the object. We take a calibration stack of a 2D target. Then we calculate one image with extended depth of field. This is just a little movie with all the images from one tiny object, just the extended depth of field images, the camera positions where the images were taken. Roughly 400 camera positions with spatial difference of 10 degrees, roughly 30 stacked images for every pose. This is an automatic process that takes 12,000 images for every object and combines them to these EDOF images. The process is fully automatic. It takes roughly 30 minutes. No human interaction needed. What we have in the end is a geometrically correct representation of the object. This is a part of the original image. This is a part of the surface model. And you see these small indentations and this small scale structure on the surface. We have this also in the geometry of the model. Extract the photographic information from these images and put them on the model. Then we end up with a digital twin. Sketchfab library is an open library. You can upload 3D data. This is a potato beetle. Maybe some of you know it. When you have a garden and you try to grow potatoes, you will not be very happy. We have this digital twin. The question is, what can we do with it? We can do, for example, measurements. Normally, when you're interested in the size of an object, you can use a scale or whatever you have. The smaller the object, the harder it is to do precise measurements. With insects or other small objects, you will try to do measurements under the microscope. And this is not so easy. We ask a couple of trained and untrained people from student to professor level, and they were asked to measure some distances on this beetle and on the 3D model of the beetle on the microscope and on computer. When they use the 3D model, the measured sizes, the variation is smaller, independent who measures it. Is it an entomologist or is it just a pedestrian walking by? They produce all the same quality of scientific data. And this is even closer to the ground truth. That's cool to use 3D models when you want to do size measurements. And also when you want to have access to measures that you can hardly measure on a complex object. For example, the surface and volume. It's easy to calculate surface and volume from geometric structures like a ball or cylinder. It becomes much harder when you have a complex object. When you want to know the surface and the volume, this is only possible when you have a digital model, a digital twin. That's what we can do with these textured polygonal surface models. They look nice, but they look not really exactly like the original specimen because there is no transparency, hairy structures and so on. They are not easy to be resolved in the 3D model. You have the advantage of having very good and precise geometry, but the visual identity of the original with the object is fine, but not perfect. This is something we can do with Gaussian splatting. This is very interesting when it comes to visualization, when you do not really need the models to do measurements, but just to show them and to communicate these models. We have the point cloud, and this is a machine learning step. Each of these points receives, first of all, a random set of size, sphere, and of colors. Then the points are compared to one of the original images and are adjusted until they look as much as possible like the original image. The next iteration uses another image from another angle, and so on and so forth. This is an optimization process that shapes the size and the color and transparency of these Gaussians. In the center, there's one of these 3D points. And this is the result. First of all, it doesn't look very much like a beetle because this is all Gaussian splats. They have the size, the transparency, and so on. But we can render this now in real time. It's a lossless PNG compression. But including the model data and so on, you will have maybe 2 GB of space that you need. This is not really a model. This is a real-time rendering. Behind this still are standing all these spheres, and it's just an algorithm that very quickly renders it depending on how I move the object. When we go very close to the object, then you can see here that these are just the splat. This is not a surface model. This is just a visual mimic of the original specimen. But it's so great because you see all the tiny hairs here as individual hairs. For the visualization, this is really fantastic. Let's compare it. This is an image of the ground truth from this fly. And this is what we receive with photogrammetry. Non-transparent wings, hairs are not visible. This here is the visualization using this Gaussian splatting. You can see these very tiny claws, hairs, the transparency of the wings, and so on. This is really a great new way of analyzing 3D data. It's not been out in the market for too long, but I'm sure this will be something we will use more and more in the next couple of years. This is an image taken at the Natural History Museum in Berlin. When they rearranged their insect collections, they have roughly 15 million of individual insects in their collections. And many of them are preserved dried and on a needle in wooden boxes. But can you see them when you go to the museum? No, you can't. Because they're somewhere hidden back doors and they're very small and not very catchy. How can we use digitization to bring these tiny objects to the public and make them attractive? There was no viewer that combines the 3D model with the actual 2D images. So this is something we developed. On the left, you have the 3D model, which doesn't have every detail, but we have the 2D images. Here you can really count and see every hair on the leg, for example, something which is not present in the 3D models. This is a very beautiful little beetle. It's roughly 6 millimeters in size. It was just crawling around at the university, so we caught it and made a 3D scan of it. And this is how we think it might be presented in a kind of a virtual museum, now in the form of a short movie, an animation. You're standing in front of the desk, there is this beetle. Now, this should somehow mimic the scanning process. You already learned that it's totally different. This looks more like Star Trek and not like photogrammetry. We have the 3D model and we can even apply a skeleton within this model, artificial bones, and we can start to animate it and bring it to life. So that this digital twin can walk around, drink some water, for example. This is a great opportunity for communication and for teaching opportunities and for bringing small objects to the public. You can put them in a 3D printer and print them out. In biology, every species has a name, and this name is always based on a single individual that is used to describe a whole species. This is called the holotype. Normally, this holotype is just stored somewhere in a museum, but we can use this holotype to digitize it. We described a new ant species from Ecuador, and this is the first species that has a physical holotype and also a digital holotype. This is how the queen of this ant species from Ecuador actually looked like. You can now either go to the museum and look at the physical specimen, or you can download all the digital information and look if you want to see something about the morphology or so. James Cook, he was traveling with his ships around the world. In those days, it was always good to have some naturalist on board. James Cook took Joseph Banks. He was a botanist, not too much into insects and animals, but nevertheless, he collected a lot of material on this first sailing around the world. And this collection he gave to Johann Christian Fabricius, who was the founder of the Zoological Museum in Kiel. We thought it might be nice to bring such important specimens, 250-year-old, back to the public. This is a 250-year-old specimen. It's a holotype. Specifically, this individual was used to describe the species. We made a scan of it, made a 3D model, did some animation, and brought it back to probably somehow how the environment looked like where this specimen was collected and described 250 years ago. That's the good thing with insects. Once they are dried and pinned in a collection, they will keep their color and shape for hundreds of years when you take care of them. Another story is Paulinus of Trier. He was a bishop in Trier and he was banished to Turkey by Constantine, and he died there in 358. But he was brought back to Trier in 395. A couple of years ago, the University of Cologne started to investigate the remains of San Paulinus. The actual question was how the textiles managed to survive this hundreds of years and traveling from Turkey back to Germany. They investigated the skull, and they found a fly puparium in the nostril of the skull. That's how it looked like. A colleague determined this fly pupae and said, this is Mya albiceps, nowadays one of the most important flies in forensic entomology. But it was just introduced to Central Europe a couple of decades ago. This was not known. We made a 3D scan of this 1,600-year-old puparium that survived in the nostril of a saint's skull. Just think about it. 1,600 years in the nostril of a saint's skull, and then you get scanned. These little dots, let's not talk about what they originally were, but now they are dried and old. Imagine you're standing in a museum and there is the skull and there's the presentation. You can now use these tiny structures and bring them to the public that are in the museum and can now interact with such small objects. We also use these models for teaching, for branded learning. For example, DigiTip, you can use the 3D models and the images as objects, and you can learn about the diversity, not only of insects, also of other animals. Paul Zorkowiak, a 3D game designer, was interested to program learning games about insects. His idea was that insects should really look like they do. These are interactive games and you play different insects and you have to fulfill tasks that actually fit their ecology. When you are a pollinator and you try to prey on something, it will just not work. This is the trailer of the game. You see these different insects, learn about the ecology, about the biology, about interactions of insects with each other and with their environment. Artificial intelligence is used also in ecology and entomology and in biology, and it's used a lot for species identification. There are huge platforms, for example, iNaturalist is a community for people that are interested in nature and they don't need to be experts. Here there are more than 10,000 observations in iNaturalist. But all of them somehow see the normal dorsal position from the backside of the beetle in its natural environment. But when I send an image from the very same specimen, but from the ventral side, the algorithm is lost. It's lost because it just lacks training data. When it comes to biodiversity, normally you will collect hundreds of insects and they will lie in alcohol in different positions with the legs somehow oriented. And there's simply no training data that can be used to identify them. Our idea was to use these 3D models, put there a skeleton, this is called rigging, and then producing synthetic training data just based on the models and covering the morphological space of this individual. With a mouse click, you can generate tens to the 100,000s of training data, which is already annotated from the very beginning. Use this to train the models to be able to identify species in such mass collections. This is a project where we develop the inclusion of synthetic training data together with the real training data to benefit the automatic identification. Even from 2D images from photos, you see how well all these deep cavities are restored in the 3D model. With this cone, you have these depth steps and you see how well the 3D model recovers all these deeply hidden structures, even though it's built just on 2D images. So it doesn't have really this 3D information, just the depth information. With that, I'm at the end and I want to thank you for listening.